So this is the role of quantitative and qualitative adaptations in skeletal muscle mitochondria on VO2 max. My name is Shravan. So what quantitative and qualitative adaptations in skeletal muscle mitochondria from steady state endurance training result in increased endurance performance? And the sample is usually young adults in these studies. So this covers, I see the adaptations, different adaptations in subpopulations, muscle fiber types, the small role that mitochondria actually have on VO2 max changes, what factors actually contribute to it, and why mitochondrial respiratory capacity far exceeds the body's ability to deliver oxygen in the first place. And so I'll be brief here, but my full paper goes into a lot more detail. So introduction, endurance sports has been refined as the sports have been more and more optimized. So the two most common ways to increase mitochondrial adaptation, steady state, so lower intensity for a long time kind of training, and high intensity interval training, high intensity short bursts. Most events do steady state endurance training, and so most studies focus on it in this field. So in young adults, steady state endurance training leads to both quantitative and qualitative improvements in mitochondria, particularly things like increased density and fat oxidation, but the effect of these adaptations on aerobic capacity is minimal compared to the cardiac, so your heart, and hematological, the blood adaptations that also occur. Um, so very important definitions. If there's one definition you take away, it's VO2 max. So the measure of maximal respiratory capacity or the maximum volume of oxygen consumed per minute per kilogram of body weight. And yeah, synonymous with aerobic capacity. So here's some more definitions. Okay. So context, mitochondrial volume density is a highly studied topic and has been consistently positively correlated with higher VO2 max. This makes sense. The logic, more mitochondria means more oxygen to be consumed for the body. As we learned, that's not exactly the case. Normalizing for other factors, it seems to have little effect. Qualitative adaptations, so electron transport system ETS capacity or oxidative phosphorylation capacity, may have a larger effect. We'll look into that later. And so there are two types of mitochondria, subsarcolemmal or mito-SS, 10 to 20% of mitochondrial volume, so smaller portion, and located on the edges of the muscle fibers. Sorry, intermyofibrillar mitochondria, 80 to 90% of mitochondrial volume, located near the muscle myofibrils, which are the part of the muscle that contracts. And these two respond differently to stimuli. We'll go over the types of muscles. This is pretty common knowledge, but slow twitch uses aerobic respiration better for endurance. In endurance athletes, it'll comprise a lot more than 50% of the muscle. But anyway, type 2A, which uses both aerobic and anaerobic, more quickly fatigable, and type 2X, or it can pretty much only use for short burst activities, uses only anaerobic respiration. So higher mitochondrial volume correlated with higher VO2 max. Study by Jacobs and Lundby showed that athletes with higher VO2 max had higher mitochondrial content measured through citrate synthase activity, which is a chemical marker of mitochondria. Qualitative adaptations of mitochondria also improved, such as using lipids as fuel, fat as fuel, and these two. Older studies show conflicting results, but the SUIT protocol is SUIT protocol is a modern procedure that gives better data about mitochondria, and this might have revealed these adaptations that previously we didn't know about. Increasing mitochondrial content may improve the reticular structure, which causes better use of fats, as mentioned before. So that's a qualitative change. Another qualitative change is size. So size can increase a lot, and so can the volume density. However, it's not exactly very important, as we'll cover later. Mitochondrial volume density does not actually affect VO2 max. So two subpopulations respond differently to stimuli. So in a rat study, mitochondrial content increased by up to 50%, but in that, the subpopulations responded differently. So Mito SS showed greater relative increases in volume, and these qualitative adaptations and greater plasticity. However, since IMF mitochondria are comprised of more of the muscle itself, its changes were still greater in total. Since Mito SS are located in the sarcolemma, they likely provide energy for membrane transport, so the outer part of the muscle, membrane transport. And since the IMF, mito IMF are located where the muscle contracts, makes sense that they fuel this activity. So why do they increase more? Perhaps because mito SS use fat as fuel better. Um, that could be an advantage in endurance sports. However, it doesn't exactly fuel muscles in the first place. Either way, it doesn't really provide a benefit to aerobic capacity. So in various fiber types, endurance is often connected to slow twitch fibers, though other fibers often grow more. Slow twitch fibers could help peripherally as they consume large amounts of oxygen, but that aside, its increased fat oxidation doesn't really affect VO2 max. Fast twitch growth, not really important at all. So yeah, no, sprinting won't exactly help mitochondrial growth, and that won't really help VO2 max. So why mitochondria aren't really important? Q peak or your peak cardiac output is far more important. Some say 70 to 85% of one's VO2 max limitation. Q peaks also had very strong connections in the past. That's the Salton bed rest study, if you want to look into that. So to explain more studies, we have to go over phlebotomy, which is the act of drawing blood. It's a common research method used to identify the effect of hematological adaptations versus peripheral ones. So blood versus peripheral, like your local adaptations. Study by Montero et al. Phlebotomy was used to match athletes' hematocrit or red blood cell count to pre-training levels, and VO2 max was found to be the same as before afterwards. So basically, VO2 max reverted completely to baseline levels. Bone et al. Identified that stroke volume was a main factor, which is how much is being pumped, I guess, basically. But that was actually caused by the blood being easier to pump rather than your heart being stronger. So hematological adaptations are very, very important. Central adaptations, so your blood, your heart, play a much bigger role. Peripheral adaptations were debated for a long time to be a big contributor, but it turns out they're not really much of a limiting factor. Two experiments by Salton showed seemingly conflicting results. One used one-legged cycling and implying that peripheral adaptations dominate, since one used a trained leg and the trained leg had a higher VO2 max than the untrained leg. However, in 1985, it turned out when large quantities of blood were delivered to an isolated muscle group, VO2 max was much higher, implying central adaptations dominated. These two studies don't actually contradict each other. In um, the first study also used an isolated muscle group, one-legged cycling, and so the trained leg, which had greater peripheral adaptations, um, ended up having a higher VO2 max because it had so much more blood delivered to it. So the results of that study could not be applied to whole body exercise, such as running, cycling, things like that. Why does the oxidative capacity of mitochondria exceed the body's ability to deliver oxygen? More metabolic flexibility. So as the body does more vigorous exercise, lactate is produced as a byproduct when the body starts needing to use anaerobic respiration, and the blood becomes more acidic, making exercise harder to complete. So when you have more mitochondria, they spread the energy load, and there are lower levels of chemical signals that usher anaerobic respiration. Your body is stressed less, despite doing a higher level of activity when you have more mitochondria. Thus, less lactate is produced, the body's endurance is better. So even though it doesn't affect aerobic capacity, big physiological justification for increased mitochondria. So to summarize, many changes occur in mitochondria, but little affect to VO2 max. Uh, However, they contribute to endurance in other ways. Some limitations of these studies: small sample. It's very hard to have larger samples in this field. because of the sort of invasive nature of the studies. And the demographics, more demographics, old people, younger children, and women definitely need to be studied more. And here are the sources and the images.